Sir, audible?
Am I audible? Hello?
Yes, sir.
Yeah, okay. And my video is also coming through?
Good morning. We are honored to welcome Dr. Subhash Narayanan, a distinguished physicist and trailblazer in biomedical optics and healthcare innovation. With expertise spanning laser technology, optoelectronics, biophotonics, and medical imaging, he has dedicated his career to developing minimally invasive optical techniques for disease diagnostics and therapeutics. For over a decade, he has worked extensively on early detection of oral and cervical cancers using point monitoring and multispectral imaging techniques, as well as dental diagnostics and antimicrobial photodynamic therapy. His contributions have led to significant advancements in clinical applications for both practitioners and patients. A recipient of numerous prestigious awards, including the National Star of India Award 2021, National Technology Award 2023, and Anjali Mashalkar Inclusive Innovation Award 2021. Dr. Narayan has been instrumental in translating optical spectroscopy and imaging technologies from research to real world patient care. Please join me in welcoming Dr. Subhash Narayan.
So, good morning, everyone. And first of all, let me thank everyone, and in particular, Dr. Suresh, for giving me this opportunity to come before you and talk about what I have been doing for the past several years. So, I hope I am audible and clear, isn't it? Hello? Am I audible?
Yes, sir.
Hello?
Yes, sir. You're audible, sir.
Yeah, okay, thank you. Let me go to my presentation straight away. Yeah. Today, I would be talking basically on the developmental aspects of a. Sorry. Yeah. Today, I will be talking on the developmental aspects related to the of a medical device, is in particular for screening and early detection of oral cavity cancers. And the device is called Oral Scan. It is an affordable and it's an affordable device, and it provides real time feedback to the users on the tissue status. It can also help in biopsy guidance, that is, in locating the most malignant site for a biopsy, and it's ideal for use in for diagnosis of cancer or early detection of cancer in resource-limited settings like in the rural villages and other places. Our company it's a startup basically. It's called Sascan Meditech, and it is incubated at Timer, that in Sridhara Institute in Trivandrum. And our company is ISO 13485 certified. Our company was founded with a mission to develop affordable healthcare devices using biophotonics and allied technologies for social impact. Biophotonics is the area covering photonic, that is optics, spectroscopy, and electronics and biology. So, about the need for developing a device is basically due to the historical incidence of oral cancer in the country. More than 337,000 new cases are detected in 2022, and out of which more than one of the burden is coming from India. And this oral cancer is caused basically due to the consumption, as you all know, of tobacco products like gutka and pan and aracanate also. And smoking is a major concern. Alcohol, along with tobacco use and alcohol, the disease can become very severe. And there are genetical causes also for oral cancer develop, as well as sexually transmitted diseases are also connected with oral cancer development. Even a non-healing ulcer in the oral cavity can be a causative factor for oral cancer. The prevalence, as I told you, is around 70,000 cases in India, out of which more than half the population end up in death, basically due to the fact that the disease is getting detected only in the late stages of its development. So, and due to that, the survival rate, the 5-year survival rate is less than 50% for oral cancer the world over. And major challenges the oral cavity cancer that is basically we use a white light of the oral cavity. White light is, for example, a torch light or any white LED light or a mobile phone also is used to examine the oral cavity for abnormal lesions. And the clinicians find it extremely challenging to locate the most malignant site for a biopsy, or even distinguishing between leukoplegia and even severe symptoms of cancer. So that's a major concern, and and there are no affordable screening tools available in the country. Especially in India, even abroad also, the kind of tools that are available are all subjective in nature, and are not available in rural areas to do the screening. So there is a lack of expertise for accurate diagnosis. Even a health worker, if you have a health worker, she finds it extremely challenging to see whether the patient has to be referred to a doctor or not from a urban setting.
Excuse me, sir. I'm sorry to interrupt, but are you presenting something?
Pardon?
Are you presenting something, sir?
Yeah.
We're not able to see it, sir.
Why?
Sir, you are not presenting your screens here, so you're not unable to see the presentation.
So 1 second, 1 second. Okay. Is the screen visible now?
Yes, sir, it's visible now.
I'm sorry for the inadvertent mistake. Anyway, shall I talk again, or
You
because
can continue.
yeah, I can continue. I think this is
Yes, sir.
about basically about the mortality rates I was discussing. That the mortality rates are in excess of 50%, basically due to the fact that the disease is getting detected during the late stage of its development. And so the survival rates, the 5-year survival rates also less than 40%. And as I told you, the challenges based are basically due to the conventional method of oral examination that is being used with the white light. And the clinicians are finding it extremely difficult to locate the grade of malignancy, whether it has to be referred to a hospital for biopsy or not, and from where to take a biopsy is also a concern with most of the specialists. And these specialists you don't find in rural setting. I mean, in a rural setting where the villages are located and people have large incidence of oral cavity cancer due to the lifestyle habits of those kind of people. And so because the lack of awareness is there, these are issues which are faced by these people. So there is a need to develop affordable screening solutions that can be used by health workers in remote settings. So it is in this context that we have been trying to develop a product that can be useful for screening and early detection of oral cavity cancers. And this is the product which we have developed. It's a multimodal imaging device with which uses a proprietary software installed on a Windows system, and also a cloud-based machine learning algorithm for assessment of tissue status. The device works basically on the principle of the diffuse reflectance, that is the scattering of light, and absorption and emission of light from the biochemical constant of the tissue. So you can see that when light is falling on the tissue, or any surface for that matter, a portion of it gets reflected from the surface, and there is a specular reflection component also based on the surface irregularities. And a small portion of the light that goes inside the tissue undergoes multiple scattering, that is Rayleigh scattering and elastic scattering. All kinds of scattering happen, and the tissue absorber that are inside the tissue, like NADH or FAD or PP9, they absorb this and emit at a longer wavelength. That's the fluorescence. It's called the fluorescence. And in addition to that, the absorption changes in the biochemical constituents can be detected from the diffusely reflected, that is the scattered light, back scattered light, which is coming out of the tissue. If we can collect that light, which will be very low intensity, then we will be able to analyze the tissue status by from the changes in the biochemical constituents in the tissue. Okay, these are the two techniques which can be used. One is the fluorescence and the backscatter light, that's the diffusely reflected light from inside of the tissue that carry information about the biochemical changes in the tissue. These are two factors that are used in our bimodal imaging device. So the detection principle is based on the absorption and emission from the biochemical content of the tissue. What we are looking at is the heme cycle here. The heme cycle is disturbed in the cancer cells. Basically, heme cycle is the production of heme. In this, all mammalian cells, in they synthesize heme by combining succinyl CoA with glycine in the presence of delta aminolevulinic acid synthase. So there's various biochemical in between constituents are there that the protoporphyrin gen 9 and PP9, and from PP9 heme is produced. These are all enzymes which convert these compounds into an protoporphyrin 9, and ferrochelatase is the iron containing enzyme Fe2 plus, in which whose presence protoporphyrin gets fully converted to heme. In cancer cell, the problem is that the ferrochelatase is low in concentration, so that the heme production is limited or reduced, and there is a buildup of PP9 in the cancer cells. So if we have a laser or a wavelength of light which is matching with the absorption of PP9, we can excite that molecule, and the emission from that, the fluorescence emission from PP9, can be monitored spectroscopically or an imaging system. So we can see here 405 excitation will give a long wavelength emission around 650 and 735 nanometer. So this is the emission of the PP9 that can be detected as and used as an indicator for malignant transformations in the tissue. Again, another aspect we are looking at is the changes in the heme production. Due to the heme production, there are two components of heme, that is the oxyhemoglobin and the deoxygenated hemoglobin. This oxygenated hemoglobin, HbO2, has a very good peak, two peaks at 545 and 575 nanometer, whereas this HP, that is the heme has a higher absorption cross section at around 620 or 650 nanometer. Up to that range, there is a stronger absorption as compared to the oxygenated hemoglobin. So when we have light sources at these two wavelengths, like for example 545, which matches with the absorption of oxyhemoglobin, where the absorption of oxy and deoxy are more or less equal, but it's a strong absorption peak. And in the case of 620 nanometer, the absorption due to HbO2 is low compared to HP. So if we can have light source at these wavelengths and collect this backscattered light intensity, we will be able to have some kind of signatures that can be used for as indicators of malignant transformations in the tissue. So all these techniques are good enough only for superficial lesion or the epithelial tissues where the penetration depth is not much. For example, this can penetrate 2 millimeter or something around that distance. So how oral scan works is basically it's a multispectral imaging is used with violet, green, and red LEDs. Violet light is to induce the fluorescence of PP9, that's protoporphyrin 9 autofluorescence, and the green is to match with the absorption of oxyhemoglobin, and red matches with the deoxyhemoglobin absorption peak. And we have incorporated a high resolution camera inside the probe itself that can capture the diffusely reflected light, that is backscattered light, and the tissue autofluorescence. And we have developed a proprietary software for controlling the camera operation, because the camera has a USB USB connector that is going to the computer, and it's controlled. The camera operations are controlled by a software, proprietary software, which is installed in the device, and the image processing is also conducted using the same software. You can see how the device works here. You can see that with one click of the switch, all the wavelengths, all the LEDs fire sequentially, and with each firing, you capture an image. So we have monochrome images captured by three different monochrome images on excitation with red, violet, green, and red LEDs are captured uh, in around 1 to 2 seconds time. This can the fast capturing will avoid the patient movement and camera shake, so that registration of the images during the image processing becomes much easier. And we have a cloud-based machine learning algorithm which will provide the user feedback on the tissue status in real time. So what is to be done is that the device needs to be calibrated to the dark light conditions, that's ambient conditions in which the measurement has to happen. Why we are using dark light conditions? Because the re-emitted light from the inside of the tissue is very low in intensity, and if you do on a normal room condition, the sensor will get bleached by the high intensity. And what we have to look at is the diffusely reflected light and the fluorescence emission, which needs a dark room environment to capture. And first the camera is switched on, and then we can visualize the oral lesion using the fluorescence mode. You can, in the video mode, I will show a demo in between. At that time you can see how the imaging is to be done, but for the time being, let me tell you that the initially on after calibration of the device, it can be used to visualize the abnormal lesions, abnormal sites in the oral cavity, the leukoplakia or erythroplakia, or the cancerous tissues can be seen with naked eye from the autofluorescence. And once you locate a lesion, you focus the device onto the lesion and capture the images. And then once the images are captured, the monochrome images, you can mark the region of interest in those images, and then the program will analyze our proprietary software will analyze those captured images and locate the most malignant site for biopsy. That is a biopsy guidance recommendation is given based on the DR ratio values represented in green, red. You can see in this bottom picture, the green denotes healthy tissue, yellow denotes potentially malignant pre-malignant tissues, and red denotes malignant tissues. So ROI is marked on the suspicious area, as you can see here, and in an adjoining normal lesion. This is for comparison between normal and abnormal. How much is the variance, the percentage variance of the ratio value? Accordingly, the device will recommend a biopsy, or no biopsy is needed, will be recommended. Accordingly, the patient can be referred to a hospital for taking a biopsy. And then the biopsy results can be uploaded into the device also for improving the algorithm as a feedback. So, and report can be generated as a PDF file and shared with the patient. And this is you can see that the biopsy results are downloaded into the system, and then it gets uploaded to the cloud, and the algorithm which manages this diagnosis is in the cloud. That's how the device functions basically. And you can see the objective discrimination using cloud-based ML algorithm. You can see that the green points in this particular graph denote healthy sites, whereas the malignant sites of higher ratio value. This is a 610 by 540 ratio, which increases with respect to the increase in malignancy. So, and we have obtained a sensitivity of 97% and a specificity of 92% to discriminate between patient normal and oral potentially malignant. So that's a precancerous tissues. You can discriminate with this kind of accuracy. And these results are published in the Journal of Biomedical Optics 2021 issue, in which Dr. Suresh also was involved in this particular study to validate the oral scan device. So the benefits, what we get out of this study is that for the patient, early detection and better prognosis. And it's a being non-invasive and painless process. Patient compliance is much better, and we can do the screening in a very short period of time, maybe in a few minutes. You can complete the screening. And for the health care providers, this provides increased diagnostic accuracy. That is, as compared to what is available right now with visual inspection, they can have an accuracy of around above 90% to detect, screen and detect oral cavity cancer. And another advantage is the enhanced workflow with cloud-based data management. So the users get an opportunity to save the data in the cloud, and whenever they want, they can download their data. And for especially in the case of monitoring of the effect of chemotherapy or any other treatments, they need to go back and see how much the improvement is there in the patient and all. This can be done very well using this repeated screenings and the workflow. I mean, the data can be retrieved from the cloud to see how much improvement is there for the lesion and things like that. Even in cases where the patient is asked to stop the habit, that is, they are frequent users of tobacco, you can tell them in early stages, you can tell them to stop using tobacco, and then they can be monitored after a month or so or every 2 months to see whether the disease is regressing or increasing, and accordingly they can take a decision on whether a biopsy is to be taken from the patient or not. So for health system, this is a very cost-effective solution for mass screening program because uh, of various reasons. This is doesn't use any consumables or similar system, and it can be done in a tertiary care setting, like in a PSC or a CSC community health center. So, and it can be clubbed with their if for mass screening there will be simultaneous screening of various parameters like the health of the patient, like the BP, the sugar levels, and all. And this can be integrated into the platform, wherein all the data gets pushed to their platform so that consolidated reports can be given to the user. This is especially useful in the case of NGOs who do large-scale screening programs. And this system can reduce the burden of tertiary care facilities, because every time once a patient comes to the tertiary care center, they do a initial checkup, and if a biopsy is needed, they take a biopsy from some site and all. In the it may not be representative of the tissue status. Sometimes underdiagnosis happen, unwanted biopsies happen. So that kind of cost and other things associated with this kind of biopsy and patient care can be minimized, and unwanted treatment can be avoided also on patients, based on the initial screening, which is done by the conventional methods. This is a demo video of oral scan.
Sascan has developed a handheld multimodal imaging device called
I hope
Oral
it's
Scan.
audible there.
Oral scan can be used in the screening of oral cancer and precancerous lesions, and also helps in selecting an appropriate site for biopsy. Oral scan probe consists of circular arrangement of 3 sets of LED lights with a monochrome camera at the center. There are certain infection control measures to be followed, like the probe has to be covered with a cling film. While wrapping the front portion of the probe, it has to be wrinkle-free.